I gather you can hear me now. I hope you can. Let me know if you can't. Okay, so we're going to start on weird happenings at the Evesham Bell Tower. But before we do that, I just want to quickly say, yeah, I'm a treasurer of 2 charities and we've had problems with the bank as well, just out of interest. Okay, so there's something for you. We've been studying for about 18 months now, 3 different eyries throughout the country. One of them was Evesham Bell Tower. I'm going to show you some video of that very shortly. But the other one was a study at FAB Peregrines, run by Natalie Mayhew, who has been fantastic for us. And we are going to be showing you a lot of information on the London one. But here we go. Have a look at this video at the Evesham Bell Tower. Oh. Sorry about the glitch. Can you see this?
Yeah, it's fine. It seems to be working now.
Good. Okay, so this is a 3-year-old tearsaw who doesn't really know what he's doing. This video seems to be stopping, unfortunately. Oh, that's a great shame, actually. Oh, here we go. Oh, yeah, okay. Oh, that is a great shame because you missed that, and I can only think this is a bandwidth problem. Trying to do Zoom as well as PowerPoint, it might be a problem. I'm going to move forward from there. Okay. What happened actually there was that the tearsaw got himself in a bit of a mess. And he did a somersault over the eggs and looked very surprised. And it's such a shame I couldn't show you that because it's hilarious. It's not what the book is about, but it's something that came about as a result of the work we did at various sites. And it was an amusing video to start this talk off. Sadly, it wasn't to be. So this is Azina and Tom. And these are the peregrines at FAB Peregrines. At Charing Cross Hospital, which is so ably looked after by Natalie Mayhew. And again, thanks to her for allowing us to get involved in this. She's been brilliant. She keeps forensic, meticulous notes, and she's helped us throughout. So a big shout out to Natalie, who's one of your LNHS members. I'm going to move this forward now and show you Azina, which is a female, 4-year-old female. And she's actually come over from the Netherlands 4 years ago and done a 375-kilometer trip to get here and is now breeding in London, which I'm going to come back through to later. So there she is with her 4 eggs, and you might notice in the foreground there's a pellet. I think you might be able to see that. Now I'm going to forward on to various stages of the process of incubation, but also of hatching. This is day 1. So I'm going to have here a sequence of days, which we managed to photograph and which for which Natalie sent us the data and the information. So this is day 1. Beautiful. Day 5, bit of a change. Day 10, a lot bigger now. Still the 3 eggs, the 3 other eggs didn't hatch, and we knew fairly early on that they weren't going to. Day 15, now look at how big this youngster was eventually called Indy. She's got very large now, just after 15 days. It's remarkable, isn't it? Look how large she is, but particularly look at the toes. those toes and the tarsus are actually pretty well as big now as the adult females. And that happens with other raptors as well. It happens with goshawks. It's a defense mechanism. They grow the talons and the feet quickly in order to defend themselves rapidly, and they delay actually growing feathers and other aspects of their biology. So now we go on to day 20, even bigger. And actually, she looks, her bill looks just as big as Zena's now. Going on to day 25. You're starting to see some pin feathers coming through down here, look. And again, she's much bigger. Day 30, now the feathers are, we're losing the down feathers here. She's starting to look like a proper peregrine. She's been ringed, look, there's her BTO ring. And the other Darvic ring, you're going to see a bit later. Day 35, she's out of the box. By the way, this box was supplied by Raptor 8, and we provided the camera for Natalie, and we've left that with her, and it's the very least we could do given the fantastic amount of information she's been able to provide us. So there's Indy now with a leg ring on, being fed by Xena on day 35. Now coming up to day 40. Now this is Tom. That's the male. He's been here 12 years now, and he's just come in with a starling, and he brought that in alive, which is a little bit unusual because they usually kill their prey beforehand. And also another item of prey was brought in alive around about the same time. We think this might be that she was going, or he was going to give the prey to Indy and let her kill it, just as one of those training things that happen when birds are at this age. I've seen this before at other sites. So this is day 40. And actually on day 40, Indy actually fledged. Okay, so this is some of the analysis that we did with Natalie. And what we're doing here is to look at how much incubating the female did as compared to the male. Now, if you look here, we go from 0 to 40 days, and the hatching period and the egg period was March the 7th to April the 16th. So the green blocks are when neither adult was incubating or brooding. So you can see egg number 1 laid at day 1 on the far left there. There was no brooding done. Well, very little actually, initially. You can see that the female gets on there from time to time. The male has a go at certain times, but only in a small way. And the large blocks of green here are when there was no adult there. And of course, this is quite normal because adult peregrines, when they have a nest of, say, in this case, 4 eggs, they'll only hard brood, hard incubate, I beg your pardon, at the 3rd egg or the penultimate one. So then at egg 3, which is there, day 6, now we find we've got constant incubating. And you can see the red samples here are the female and the blue is the male. Now, when the female goes off to do whatever she wants to do, to have a bit of a fly around, etc., then the male will come on straight away. And you can see going across the period here, there's a defined, almost a very defined period of time when he takes over. She always incubates throughout the night without fail on every single night of the incubation phase. He comes in at around about 6 o'clock on average, and then he has another session here at 6 o'clock in the evening, and occasionally, as demand requires, he'll go on. Now, what we calculate is that the female is on the eggs for 84% of the time, the male 16%. And this is really quite standard. So thank you, Natalie, again for that. You'll be hearing me thank Natalie a lot in this presentation, I think. Let's now look at hatch to fledge. Now, what we're doing here is, again, the same color coding. So the large blocks of red are the female. She then is brooding fledglings at this point. And you see on day 2 here, the male is brooding. And he broods there for 30% almost of that second day. Now, in my experience, this is quite unusual because often the female will be very jealous of brooding her youngsters. And you won't typically see the male brooding at this frequency that early on. Now, I think this is probably something to do with the Xena being 4, Tom being 12, Tom being an experienced breeder, is more likely to know what he's doing and to be able to take over satisfactorily. In another eyrie we looked at as part of the study in 2022, we found that the male was not allowed anywhere near the young until day 8 or day 9. Now, when we come to feeding, we have a situation where on day 26, the youngster starts feeding itself. Indy starts feeding itself. And then by day 31, he's feeding himself regularly. And then forward to day 40. So the chick is feeding 15% of the total time, the male 10%, the female 75%. Moving forward. Now this is the number of prey deliveries by time of day at Charing Cross Hospital in 2022. And what we've done is to assess when these prey deliveries are coming in. And by prey delivery, I don't mean an intact prey coming in and being delivered. I mean individual prey deliveries of part prey sometimes. So you'll get a pigeon coming in and then it will be fed to the brood, maybe 3 or 4 feedings. So it's these partial feedings that we're talking about when we talk about prey deliveries. And what we see here is that there is a period of time right at 5 o'clock around the early part of the day, which typically across the whole of the period of prey being delivered, she'll come in and feed, or he will come in and feed at that particular point and deliver prey. Then you get a breaking off of that in the middle of the day. And then again, a little bit here between 1 and 3 o'clock, a little bit more prey coming in. But typically, and again, this is typical of the other sites we've looked at, we get this large period of time here between 5 and 8, where we're getting a lot of prey coming in, and then that prey is often cached. So the average number, this blue bar across here, the average number of prey is around about, that's prey deliveries of a particular element of prey, is around about 9 to 10 feeds per day. Okay, so what were they feeding on? Well, we've got 93 different prey items coming in 2022 at this particular site over about 40 days. And 86% by biomass was pigeons. 6% was starlings, and 5% was ring-necked parakeets. Now, of course, that is only a recent phenomenon, and I believe that parakeets are growing in their population. I'm sure that the LNHS will understand, to what degree they are, but clearly that's a significant portion of the prey. I mean, clearly also the main element here is pigeons and doves, including racing pigeons. So this really comes from the data we could see on those recorded cameras, and also particularly from Natalie's meticulous notes. And that is Indy, PDT, with a young green woodpecker that was taken in Regent's Park. And that was just a few days after, not after she'd fledged, but after she'd sort of disappeared and the adults weren't feeding her quite so much. And we felt that she might be able to cope on her own and feed for herself. So this was probably one of the first prey items she actually caught for herself. It's a remarkable image, and it was caught by a guy called Warren Richardson, I think. Natalie will correct me if I'm wrong there. And we think it's a fantastic shot, and it's wonderful to see this away from the eyrie. And this image is in the book. Okay, I'm just going to have a bit of a glass of water. Now, what we've found here is in 2022, Koei Tanaka and Dala Zelenitsky in southern China found this collection of dinosaur eggs. Now, each of these eggs, and I don't know whether you can see the arrow on here, can you? That I'm putting up against the PowerPoint. If you can't, each of these eggs is around about 9 inches long. And you can see there's a pod of the eggs. We found out actually that these eggs had been hard incubated clearly and were about to hatch when this happened. So if we carry forward with this, now this is in the book, and this is to do with dinosaurs being ancestors to birds. So this is one of the eggs, and it is thought it's an oviraptoroid theropod from the late Cretaceous. So it's 66 to 100 million years ago. And then having got to that point, as you saw in the nest there were 10 or so of these, about 9 inches long. So if we then go to the next slide, it's cut open laterally. And that's what was found inside. Now, this is the bill here, the cranium, and then the skull, the skeleton, humerus, and various other bones here. Then they did an X-ray of that just to see what was underneath, and particularly the way that configured. And I'll show you that now. Now that's essentially a cleaned up version of the X-ray. So you can see the configuration here, which is not dissimilar to a pigeon at this stage of incubation. It's clearly almost ready to hatch. And then an artist's impression of that bird. Remember, this is some 60 million years ago. It's quite revolutionary actually to find this in China. Just before we published the book, actually, we came across this piece. That is a pigeon, which is not, I mean, you can't see it adjacent to the previous image, unfortunately, and it's in a different plane. But if you look at it side by side, you'll see that there are a lot of similarities between this and the previous reconstruction based on the skeleton that was found by taking X-rays. And this is an artist's impression of that nest and what that bird might have looked like. Of course, you know, it is an artist's impression. It's a theropod, it's got 3 toes, we know that because of what was in the egg. That would be, I think, 1.8 meters. I've done the calculation, it's in the book. I'm talking off the top of my head now, but that's about 1.8 meters long, that bird, the parent bird there. So we thought this was a really interesting thing to find, and we found it right at the end before we finalized the book. So we've included this and, of course, a lot more besides on that particular subject. Now, I'm moving forward now to this. Now, these, Richard and I went to Tring to have a look at the Tunstall peregrine, which is the definitive peregrine falcon in 1771, labeled by Tunstall. But we found, as well as that, when we were looking in the drawers for the Tunstall peregrine, and there are photos in the book, we found these sakers. And we asked the lady curator there what this was. So these are 4,000-year-old sakers, which were votive offerings to the gods. And they were apparently mummified and produced on an industrial scale. Hundreds of thousands of these were found, and they traded them in what was Egyptian, but it's Egypt. They were traded and used as votive offerings for the gods Thoth and Horus. Now, you'll recollect that Horus is a god of the sky, and he was actually a man with a falcon's head. In X-ray, these birds were found to have reeds and all sorts of detritus in them. One had a third tibia. So the traders of these weren't particular about the makeup of what they thought, what the buyer thought would be a votive offering to the gods, being a mummified saker. And that is something that was written by, that is not just me surmising, that is written by the curator at Tring, who's an expert in this particular area. This is an image that I want to show you. It's similar to the front of the book. But the the thing about this is, if you look at the juvenile on the right, it's almost as if it's standing up. And that juvenile, there were 3 in this brood. This is at Simmons Yacht, which is my study area for 40 years. This is the adult coming in with prey back here. And you'll see that this particular juvenile on the right really wanted to get at the food. So if I now morph this into the next image, you'll see he fell flat on his face, didn't get anywhere near it. And this particular bird here got the prey. So that was a nice little sequence. The full sequence is in the book as a sequenced, as several sequenced photographs. And on the cover of the book, you'll get something similar to that. These are images taken of flight coming in. One is on Lincoln Cathedral. The other one is an eyrie, one of our other study areas in Scotland. And what I wanted to show you here is actually an interesting composite of itself. This is in the book again. But you'll often hear it said that the alula, which is the thumb, if you like, the alula feathers here are an aid to landing because they disturb the airspace and stop the bird from stalling. And that's a really good example there of the alula coming out. And there, at this particular stage of landing, this is another landing where you can see it here and the bird coming in there. Okay, so now we are coming to what would have been Richard's phase of this talk. And Richard, as you heard, is an astrophysicist, a physicist, and he is incredible at working out flight geometry and doing the underlying calculations of how birds fly. I am merely a stand-in for him in this respect, but I'll do my best for you to explain what's happening here, but I won't be able to talk in the technical terms he could have done. Most of you listening to this will have seen falcons in stoop. Well, what they tend to do is they'll wait on very frequently high up, maybe for the sake of example, 1,500 foot. And then if they see a bird underneath, they will power fly, and then suddenly they'll drop down and they'll come into this sort of shape at the beginning of the top here, and then form an M shape. They've still got control of the wings and their flight here, but then they'll come into what is called a teardrop stoop. Now this is a cup type flight. As you see on the right, the 2 pictures, top and bottom, show, actually it's a fantastic image, which is done up in Derbyshire by a friend of mine. And you can see you have this teardrop shape, and the wings tucked right in there, and the cup is formed. And that enables it to have as little profile against the atmosphere as possible and to reach maximum dropping speed, because all the weight on a peregrine is in the pectoral muscles at the front here. And with that cup shape, it's like a diving bomb. So it maintains that, but it doesn't maintain massive amount of control at that point when it's in stoop. But what it does do is it stoops very, very quickly to gain speed and momentum. So then it will come down on the flight, and typically you'll see flicking, a quick flick of the wings to adjust course slightly. And then at the approach phase, you'll get the M shape again when it's got much more control of the surfaces of the wing and the tail. And then it will come down and gradually slow down. And at this point, at contact, it will brake heavily, and it will brake, splaying the tail and the wings, and virtually stand on its feet vertically and take all the energy and all the speed out of the flight, so that when it hits its prey, the closing speed might be very minimal compared to the sort of speeds we get up to here, you know, the 100-mile-an-hour plus speeds here. It will be running at around about closing speed of probably 15 miles an hour in closing to that. Now that pigeon, if it's a pigeon, it could well be going 65, which is about the top speed of a racer. So this bird at that point might be doing, say, 80, for the sake of example. So that is how they'll do that. That is an anatom at the start of the stoop identified in that previous image, and that shows you the shapes. It's done by Will Suter from America, who helped us greatly actually with the book and a lot of the images he provided for us. And he's got a fantastic study site going. He's been doing it for what, 10 or 15 years now. So that's the start of the stoop. This is the cup shape, a large image there, which is in the book by Tony Fallon from Derbyshire, actually. And he took that, I think it was this year, and it turned up on Twitter. So I contacted him and asked him if I could use it. And he was gracious enough to say yes, because it's a perfect representation of the bird in full stoop and in teardrop. But he stopped it so well, because this bird would have been going very, very fast indeed. And this is one of the best images we could find in that particular shape. Okay, so this is some work that was done by Lagerman et al. 2018, and we spoke to, or Richard actually spoke. This is technical stuff, which is actually normally beyond my remit, so forgive me if I'm not doing particularly well on this, but I'm trying my best as a poor substitute for the man himself, Richard. With thanks to Erwin Gowrie, who we're speaking to. And what we have here is a gradated scale of where the stresses are on the wings at various stages of flight. So what we see, and you can see the bar here from low to high, high shear stresses on the front of the wing here, look, in that phase, going slowly down to the back of the wing here, the purple. And again, I'm not sure whether you can see the arrow on the screen, but if you can, I'm highlighting the colors. But then, and notably, and subtly, I think, what we have is green markings on the edge of the inside wing and the outside wing here, which is sort of median stress, but also on the outside of the tail in the M shape. And there you again replicated there, which gives you a control, a degree of control of flight, given that you've got pressures coming in against the wing. Here, in full teardrop stoop, we have the stresses again here on the humerus, on the front of the wing there, but reducing down. But you're not getting any of these little sparks here of pressure on these areas, because actually there is no controlling surface here. It's simply done to get the bird to its prey as quickly as possible, and to use its mass to the best effect. This is the humerus, and Richard's done a big study on humeri, and he's about to write a paper on this. But essentially, the humerus is the bone that takes those forces at the front of the bird. And you can see that it's strutted. It's hollow because it needs to be, because the bones can't be too dense. But it's strutted like this at the front edge to give benefit to the structure against pressure coming from, in this case, from the left, i.e. from the frontal area. But it's also ridged, and that stops stress bending of the humerus itself. So you've got strength internally, but you've got strength laterally. So if you try to, the bottom here is simply a toilet roll, actually. But you can see the way it's made, and it's got these structures here. So it's actually a trapezoid, I think. And it's put together in that way to maintain the strength of the roll. If you just had a roll, you wouldn't get strength bending torsionally, and it would tend to collapse. But here it doesn't. And that's exactly the same process and reason for the humerus having these ridges and being strutted like that. These are humeri of 4 falcons in the UK. So we've got the 4 predominant UK falcons. So peregrine at the top, then we've got hobby, then kestrel, and then merlin. And clearly and obviously, because it's a bigger bird, the peregrine structure of the humerus is much greater, much bigger. But if you reduce the size, taking into account the mass, then still the peregrines is stronger because it needs to withstand much more massive forces when it's gaining those high speeds. The weakest one of the lot is the kestrel, and that would be if you prorate it down to mass. The kestrel is much weaker comparatively, and that is because it doesn't stoop. It hovers, certainly, but it doesn't stoop. The merlin does, and the hobby does to a degree, but none of them to the degree that the peregrine uses it for foraging. Okay, now if any of you out there know a great deal about peregrines, you will know that the top speed rated for a peregrine in America was 242 miles an hour. But in fact, this bird was dropped from a Cessna or variously a hot air balloon, depending on which study you take, in very thin air. And it was done at 17,000 feet, which is somewhat over 5,000 meters. So what we've done is we've said, okay, at the atmospheric pressure at 5,200 meters, what is the implication of that speed if we convert it to the atmospheric pressure at ground level? Because clearly, if you drop a bird from a Cessna at 17,000 feet, you've already got forward momentum, which can translate to downward momentum, as you know, simply because you're in a plane. But you've got as soon as you drop, you will be in very thin air. And clearly then you'll go much faster because the pressure running against you is less. So if we now say, okay, if it does 242 miles an hour at 5,200 feet, what would be the equivalent MPH at ground level with the air pressure at ground level? It turns out that it's 183 miles per hour. Now, Rich has done the calculation, and I leave him to do stuff like that. But as a result of that, what has happened is we've been in touch with National Geographic and also with Guinness Book of Records, and said that we're not happy with the record being placed at 242 because it's in circumstances that are not empirical, i.e. they're not circumstances that will normally be found by a peregrine foraging at 1,000 feet, for instance. They have then taken that record out. So they have discarded that record and reverted it back to what is a more realistic figure of around about 200 miles per hour. This came directly out of the studies that Richard did. I can take no credit for that other than suggesting to him that we ought to make that conversion. Okay, now this is interesting. This concerns the baffle in the nostril. And as you can imagine, if you're doing 150 miles an hour at full stoop, what you don't want is the air to go straight through your nostril and into your lungs, because that would be very damaging to your internal organs. So it is often said that the peregrine has a baffle at the front end here of the nostril, which the jet engine was based on, and that is true. But to get a full idea of what the baffling is, a scientist has actually taken off, sheared a skull of a peregrine and taken it off along the line of the nostril up through the skull. And we can now see the internal arrangement post-nostril, which is here. Okay, so on the top left, you see the dotted line is where the cut was made. I know it sounds a bit gruesome, doesn't it? But it's where the cut was made in order to understand what the baffling arrangement was internally to the skull. This is the image of that cut through the skull. And you can see that the nostril is here. And look at all this arrangement within the skull to stop the air pressure going directly into the lungs. There's a series of maze-like cavities which the air goes through before it comes into the bird's lungs, and thereby stopping any large pressures arising in the lungs. And we found this, and we knew that it must be the case, but we found it very recently. And again, this and other biological aspects of the bird are in the book, and this is quite new piece of work that's been done. So we hope that we have brought the biological science underpinning peregrines up to date in this book. Okay, elements of peregrine vision. Now, that's a beautiful picture of a peregrine falcon, I'm sure you'll all agree. And it's all about the eye. And I want to describe certain elements of vision to you on the basis that I have an overview that peregrines, based on personal experience, can see 10 times what we can see. Science has never underpinned that and has always poo-pooed it. And we wanted to try and understand if we could get some understanding of what that ratio might be by looking at the detail of sight, which we've tried to do. Now, I must say before this is very much Richard's area. I'm going to cover as best I can for him. Forgive me if it's not as it would have been, but I'll try my best. So vision is made up of various elements. One of the elements of vision to measure acuity is called cycles per degree, or CPD. Now, I want to explain that. So here's the eye, and you have a 1 degree of arc that you isolate, which you can see. And if you've got 1, 2, 3, 4 black bars and 4 white bars, then at 1% of arc, you would say that this eye could see 4 cycles per degree. So a 1 degree of arc, 4 cycles, i.e. a black and a white, 4 times. So that will give you a 4 CPD definition of what that eye can see. Right, so having understood the basis of CPD, we can then start to compare CPD for humans and for other organisms. So the human CPD would be 30 to 70, and as one gets old, you lose that facility. A young man or a young woman would have probably 30. Sorry, 70, I've got that wrong. Sorry, 70. And an older person like me, 30. So Martin from 1986 postulated that large falcons have 2 times the visual acuity. So maybe up to 120, 140. Raymond in 1985 measured by test a wedge-tailed eagle, and that was proved to have multiple tests, by the way, of 132 to 143 CPD. Now, this is a spatial measurement of visual acuity. So if you take the 70 here for the young person, compare it with the 140 here, you can see that in respect of cycles per degree, a spatial measurement, then the large falcon or the peregrine, in fact, has twice the acuity in spatial measurement. So, okay, that's a starter. And that can be done by test. And then we come on to something else, which is temporal vision. Now, we're talking now about fast vision or slow vision. Let me explain what's going on here. So on the axis, the upright axis here, you've got FFF. That's flicker fusion frequency. And that ranges on this particular chart from 0 to 125. Now, let me explain for those who don't know what flicker fusion frequency is. If we take humans, then your television will be at a frame rate of something like 30 frames per second. Why? Because the human eye cannot distinguish between seeing 30 frames in a second and a video. So that's how quick the human eye can work. So use that as your basis point. 30 frames per second is what a human can see. So he can judge that he's got a video, not 30 individual pictures coming in front of him in 1 second. A neon tube flickers at 50. So if some people here in the audience see neon tubes flickering, then they can see a 50 FFF rate or flicker fusion frequency. Some humans can, some don't, some are bedeviled by it, some aren't. The Harris hawk at a luminance of, let's talk about this luminance on the bottom line before we go any further. The luminance is a statement of brightness. So obviously the higher up here, the brighter it is. Now, peregrines love bright days. So let's talk about this line here. Well, let's talk about this line here, 40,000 luminance. The peregrine has a maximum flicker fusion frequency of 129 and an average of around about 119, 120. The Saker is down at 100, the Harris hawk is down at 75, which means that if you take the human rate of around about 40 and you take the peregrine at 120, then the peregrine sees faster by a factor of 3. What that means is you can see 3 times more detail than we can, close to and far away. So there's another element of sight which is significantly different in a peregrine. What we think happens here is the flicker fusion frequency of searchers and long-range hunters, such as a peregrine will get high and it will be hunting all around, looking all around. It needs to see objects that it can prey on miles away. Clearly, it benefits the peregrine to have a very high flicker fusion frequency, and it has been tested as doing so. So there's another element of the comparison between human and peregrine. Moving on, there's another adaptation that birds have that is different from humans. And that is the physical structure of the eye itself. So with humans, we have the Brooks muscle, which moves the lens, it acts upon the lens. You know, when you squint, you want to see something. As you get older, you tend to squint more, I find. You use the Brooks muscle to squeeze the lens to see things you can't see with the eye at rest. So, yeah, peregrines have that. But importantly, very importantly, they have the Crampton's muscle, which acts on the cornea itself. And it pushes, the Crampton's muscle here, look, is the, if you can see it on the screen, is the blue element. And that pulls in the cornea. And it can move the cornea up to as much as 28 diopters in red-tailed hawks, 16 diopters in American kestrels. We could not find any analysis of what the diopter might be for peregrines, but it is not unreasonable to assume it's going to be in that range. What this does is it gives the peregrine telephoto capability, particularly in the deep fovea. So again, that's another adaptation that birds have over humans in terms of sight. Here we have an analysis, again, there's more in the book on this. This is just an element of it, but the deep fovea here is angled at 40 degrees and it goes out laterally. So the peregrine will see laterally, using the deep fovea, it will see telephoto because it can adjust its lens, it can adjust the cornea, and it has a very deep fovea, which means it is more acute. It's like the retina in your eye. The fovea is in the retinal area, but it also has a shallow fovea. So the shallow fovea will give it binocular vision forward at the same time. So raptors, and particularly peregrines, are twin fovea, whereas we are single when we tend to look forward. So, but the peregrine can do this simultaneously. So it's looking far right at 40 degrees. It's looking left at 40 degrees with high power vision, telephoto, it's sometimes called, but at the same time, it's looking forward as well. So its arc of acute vision is much greater than ours. I wasn't supposed to go back there, and I won't. So that's another adaptation that peregrines have to enable them to see the horizon much better than we could from their position. And this is a cross-section of the deep fovea, the top, and this is at 1/1,000,000th of a meter scale. So it's a very small area, but nevertheless very significant. As a matter of fact, scientists even today with modern equipment cannot exactly understand what is happening here and how it works, but you can, through experimentation, understand that the peregrine's view from this fovea is much better than from the shallow one, which is represented here. And this was got together in consultation and in speaking to Mindaugas Mitkus, who is an expert in the field. Now, I'm going to duck out of this a little bit because this really is Richard's area, and frankly, it's, I wouldn't say it's beyond me, but I understand it. So these are the rods, and the only point I want to make is that whereas humans are trichromatic, we see in 3 colors, raptors, well, peregrines are tetrachromatic. They see in 4 colors. They see in the UV. Now, some of you have read about this, or some of you who may know about it. So there's some discussion about whether or not raptors can see in UV. And this discussion has changed over the years. And there is a body of people in the science community that think actually they don't. We still believe, and we're by no means expert in this field. We can only rely upon others to tell us, but we still believe, based on the balance of science out there, that they can see to some degree in the UV, which will confer greater visibility of certain aspects of their environment. Now, the point to make here, though, is the cones are the ones that give you daytime vision. So for diurnal raptors, you need a lot of cones. In a peregrine, the cone density is about 400,000 per square millimeter. In a human, I believe it's around about 100,000. So, but that varies between individuals, and no doubt it does with raptors as well. But again, you see that there is a large benefit for a raptor in the density of cone receptors in its eye as compared to the human. And that's another area where the raptor benefits compared to humans. So I'm trying to now to build up a picture of what benefits structurally the raptor, the peregrine, has in its eye compared to humans, so we can get some kind of feel for what the degree of improved vision they might have. No one in the science community has come up with a number, and because they just cannot understand how all these aspects correlate. Okay, here we have the nictitating membrane, a superb image by Peter Christian from The End of Man. And you can see the nictitating membrane there. This comes in from the nasal side and moves across the eye to get rid of dust and debris in the air when it's stooping. Clearly, you don't want to stoop onto dust at 100 miles an hour. But they also have a lacrimal gland and a Harderian gland in the eye as well. And the Harderian gland will give rise to a sort of quite a high viscosity fluid, which doesn't tend to dissipate as much as the lacrimal gland. Now we're going on to Iceland, which is very much Richard's patch. And I'm going to move forward from here because I know I'm running out of time. So this is a gerfalcon in Iceland. And Richard saw it doing a flight path, which was a logarithmic spiral, and wondered why. But he didn't know at that point that it had twin favere and one offset to 40 degrees. So what he found is on a long distance flight, the peregrine will go into a logarithmic spiral. It won't go straight from one point to another at long range. And he wanted to understand it. And part of the understanding of that was to understand the science of the eye, which we outline in the book. It is not proved. This is some detailed work that was done by Tucker. And this is 1,000-meter distance here, look. And these are flight lines of individual flights to a lake. And you can see the way they're bending to the left. And this red line is the logarithmic spiral that you'd expect. And then we think at about 300 meters out, it converts to binocular and goes straight. The logarithmic spiral fits very well to those flight lines. And now, we used Mouse. This is a bird that is a falconry bird. And we used him to try and replicate these logarithmic spiral lines. And he did so twice, but it wasn't significant enough, sadly, scientifically, for us to be able to take the data. But there is a sad corollary to this, and that is that unfortunately, Mouse was electrocuted this week when landing on an electricity line. A very sad, lovely bird, and that's his owner, Oliver Tomlinson there. Now, I am aware that it's 8 o'clock. I do have other slides to show you, but I imagine that you might want to, because I've hit the time now, you might want to ask questions, but if you want me to carry on a bit longer, I'm very happy to do so.
Do you want to just carry on for a few more minutes? Okay. And then there are a few questions coming through, but maybe if there's a couple more things you'd like to share with us.
Yeah, there are. Could you just shout me out when you want me to stop then? And I'll just carry on until you let me know.
Yeah, give it 5 minutes or so. Is that something like that? Would that be all right?
Absolutely. Yeah, absolutely. I'm going to run through these quickly. These are images from our Scottish quarry site. And sadly, there were 3 chicks at this site, and the last one here that you see at the back was a smaller runt. And sadly, that particular bird didn't make it. It was used as falcons and all animals will do. It was fed to its siblings. That's a message that for Richard, because he was going to do the first part of the show, but he's not here, so I've done it. And he was handing over to me, but I'm handing over to myself now. Just for question. This show relative size. And that was the way they used to come onto the roof and down into the eyrie. They used to skid down there, and they did it all the time. So we cover all of the subspecies, and I'm going to run through this much quicker than I normally would, because I want to show you some of this in the next 5 minutes, 4 minutes I've got. So we cover all the subspecies, and we look at what the population is in each area, how big they are, what their physical characteristics are, and what their prey spectrum is. So just as a feel for this, so pelei are falcons subspecies on the northwest coast of America, essentially in the Aleutian Isles, but also on the Commander Island, which is Russian. But here in this particular area, you've got pelei, anatum in yellow, and an overlap of anatum and tundrius green, and then tundrius blue. So you've got a lot of subspecies in a small area. Coming on to pelei, this is a juvenile pelei. And pelei are the biggest of the subspecies, typically on average, with the female averaging around about 1,201 grams, as you see, the male 894. This is a photo by Nick Dunlop. For the sake of a comparison, Ratcliffe will tell you that the female average peregrinus is around about 1150, but the male is somewhat less at 700 grams. So this is a rather bigger bird on average than our bird, our peregrine, our nominate bird. This is another pelei, but this is just a quick indication of the prey spectrum. So you've got a crested auklet and a bufflehead, but this is a pie chart of the various items they take. And here you'll notice that the green, the auks and petrels, are about 70% of their total prey, and then the yellow, ducks and grebes. So this is very much a sea-going bird, and most of its prey is water-based. So we do this for all 19 subspecies, so you get an idea of what they're feeding on, how big they are, what their physical characteristics are, and quickly running through. Now, this is Peregrinator from Bangladesh. This is somewhat smaller than Peregrinus at 695 for the female. Now, that is really the size of a good Peregrinus male. But look at the coloration on this beautiful photo. And it's quite muted, those red stains here, nice barring underneath here. But that is in Bangladesh. If we now go to northwest India and look at Peregrinator there, we have this, very, very different. Very rufous. This is a male at 450, potentially grams, maybe a bit more. Male adults, look at the lovely black head and the full cap, rufous underside, with very little barring here. Again, that's a typical Peregrinator from that area. And then, and I am mindful that I need to stop, and I'm going to really run through this, but this is the prey spectrum of habitat types in Fiji. So this is Nesiotes, and you can see in the rainforest, 90% of the prey are fruit bats. Now that is counterintuitive if we think of a peregrine as a bird-eating raptor. Of course, it's not much different from a bird, is the fruit bat, but nevertheless, it's a massive proportion. So basically, they take what they can wherever they are, and that's why they're such a cosmopolitan species. That's why they do so well worldwide, because they've got a very plastic prey spectrum. Right, I'm going to quickly, this is Macropus. I'm going to quickly run through this now and I'll just show you that. I'm going to run through that. This is a nestai, which is a smallish medium bird in Malaysia. Look at the lovely black rufous and the even blacker cap there and heavy barring underneath. I'm going to run forward on that. I'm going to finish with this, which is Exeter buzzard cyclostutes, which we've done a lot of analysis on. This is about a minute and a half. I'm calling it a day there, but what I would say just as a finale to that particular video, there were 22 cycle stoops there, and these birds were buzzard haters, and they used to, as a matter of preference, kill buzzards all the time. In 9 years, they downed 470 birds to our knowledge. Sorry, they attacked 470 birds and they downed 70 over Exeter city centre. So I'll leave it at that. There is other stuff there in the book. This is a very small percentage of what's in the book, and I hope you enjoyed it. And I'm very sorry for the technical mix-up at the beginning. Thank you.
Thank you so much, Steve. That was absolutely fantastic. And I'm sorry we had to kind of call a halt to it, really. But, you know, it makes me sort of think maybe we should think about doing a second sort of slot sometime in the future, because there's obviously a lot more to cover. Do you want to just say a little bit about your book, you know, like the name of the book, who's published it, that kind of thing? Just obviously, because I think a lot of people will be interested in reading it after you've kind of whetted our appetite.
Well, thank you very much, Marie. Yep, it's called The Peregrine Falcon. It's published by Snowfinch Publishing, and it will be available from next Tuesday at Raptoraid CIO website and at NHBS. For those on Twitter, I'll be tweeting out all the details and emailing everyone I know, including LNHS people. I'm very happy also to email you, Maria, or anyone you nominate at LNHS with all the details. But so it's being printed as we speak.
That's brilliant. Yeah, no, do send something through to me, and I'll kind of share with the London Bird Club, you know, kind of like share it around. But really, I just, really, yeah, so thank you so much. It looks like it's just going to be amazing. It's just so much kind of research, and it's like, it feels like it's sort of cutting edge, you know, deepening our understanding of these amazing birds. Lots of people saying, you know, thank you for such a wonderful talk. Just the footage, the images were fantastic. And I was just, you know, kind of bowled over by the thinking about the kind of biology of, you know, how their bodies actually work. Things like the sort of bone, what the bone structure contributes, and the kind of, you know, they're deepening the understanding of their kind of visual quality of their visual perception in so many different ways. It was absolutely fantastic. We have got quite a few questions that have come through, and I'm conscious we probably don't have time to ask them of you. Could we possibly pick up the questions from the chat and email them to you? And
of course.
Maybe I mean, not when you know, only when you've got kind of got time and after you're done, but maybe kind of like do it if you could sort of pop those back to me, and then I can circulate them around to everybody who's attended. So would that be possible? Would you mind doing that?
No, not at all. Very happy.
That would be fantastic then. So we'll just kind of think about maybe inviting you back. I do want to say also thank you so much to Natalie, and thank you, she's obviously has been on the call, and thank you for acknowledging her work, because it's lovely to see the kind of London peregrines featured, and you know, it is amazing. You know, we're very impressed with the quality of the work that she's kind of contributing as well. So thank you for acknowledging that.
Can I just underline that she's been absolutely amazing. I couldn't have wished for a better person to help us out with this book, and the book is so much better with her input as well.
So thank you ever so much to everybody for coming along. I hope you've all enjoyed the session. I can see actually from all your comments in the chat that you really have. Thanks once again to Steve, and also please do wish Richard Sale well for us, and kind of, you know, hopefully at sometime in the future he'll be able to kind of join us as well. But